Thank you, Karen, and thank you everyone for attending today's webinar. All right, now let's try to get the slides up again. There we go. The objective of today's presentation to discuss the biomarker development process and also talk about some of the common pitfalls and how one would go about mitigating them. The first step when we talk about biomarkers is to define what they really are. The biomarker working group calls a biomarker a defined measurable indicator of a normal or abnormal biological process or responses to an exposure or intervention. That is quite a mouthful, but what does it really mean? In the cases that I'll be presenting today, the biomarker could be on mRNA-based, protein-based, metabolomics, or any type of omics-based, or it could be even clinical variables such as heart rate, blood pressure, or others, or it could be a combination of any of those. Now let's talk about the biomarker development process itself. The first phase of it is demand analysis, which really means to talk to clinicians and experts in the area that you're interested in to assess what are the currently unmet clinical needs, and then talk to health economists from early on to figure out if such biomarkers can be developed that have socioeconomic value. The next phase is content development, and this was covered by the first two seminars in this series, so I'm not going to talk about them too much, just to say that in these phases we discover the biomarkers and then test them in additional cohorts. The majority of this validation and qualification phase will be covered by Jiang, namely the analytical development, where the markers are moved onto a clinically relevant platform, as well as the clinical validation, where the data from coming off of this platform are tested for sensitivity, specificity, and other characteristics. The last step before clinical use is clinical utility. And this is usually performed in a way of a clinical trial, where potentially there are two arms. One arm has the patients treated using the biomarker test, and another one using the standard care. And at the end, outcomes from those two arms are compared. And hopefully, it will show that patients treated using biomarker-based clinical care are doing a lot better than those with the standard care. And the last step is clinical use. And you may think, oh, finally we made it, but there could be a lot of issues around this too, because it then involves commercialization, you know, all that's associated with it, including marketing and other ways of getting to clinic. It involves reimbursement, talking to, you know, ministry, figuring out if they would be willing to use this in a clinical setting. How did we get started in the biomarker development process? Well, the year was 2004. Yes, 16 years ago. We started with a project called Biomarkers in Transplantation. 4 years later, we had added 3 additional disease indications. With collaborations with Dr. Singh, we added the COPD biomarkers. In collaborations with Dr. Andy Ignashowski, Mustafa Toma, and some from Dr. McManus, we had several programs going in heart failure. And with Dr. Adira Levin, we had added also CKD, chronic kidney disease. 4 years later, we also had programs in allergy and asthma, mostly led by Dr. Tebet. And by this year, we now have a few additional colorful pieces to this circle. And that is due to the fact that our biomarker development process is disease agnostic. So it could be applied to any type of disease indication, from spinal cord injury to vaccines or stroke or other disease. And a lot of these programs were done with collaboration, in collaboration with PIs from HLI or other institutions. Also, some of them were fee-for-service based with pharma or patient advocacy groups. So once you start thinking about developing biomarkers, one of the first things one has to figure out is what is the fit for purpose of the biomarker. And as I was looking for a definition of this, I found this as being well suited for its designated role or purpose. And so maybe the definition doesn't say too much. So I figured we could go through together a few things that one would use to assess the fit for purpose. So first one would be, what is the question? And this would happen during the demand analysis, as I mentioned, to talk to clinicians and experts in the area, figuring out what are the unmet clinical needs so that you define the question. The next would be, who would use it? Would this be used by family practitioners? Would be used by tertiary care providers, or maybe in the emergency department? Which are the patients who would benefit from it? And how often would the test be used? Let's say for a chronic disease, you may want to apply it multiple times. And what is a suitable performance? And so ideally, you would want a biomarker that is 100% sensitive, meaning every single patient with a disease would be diagnosed correctly, and 100% specific, meaning all of the people who do not have the disease would be also classified correctly. But in most of the complicated disease that we're studying, this is virtually impossible. So one then have to figure out what is the most ideal situation. Should it have a high sensitivity or should it have a high specificity? So for example, if one wants to diagnose cancer, you want to make sure that patients who have cancer are actually diagnosed correctly so they can be treated and get better. So that would mean a high sensitivity test would be ideal in that situation. The last arm in this sort of series of questions is the cost effectiveness. Does the biomarker improve quality of life? Does it reduce cost? And so these are the questions and the balance that you have to work out, potentially working with health economists. As I was thinking about the different challenges that one could encounter within this biomarker development process, I realized that many of them could be categorized in 3 groups. So the first one of these being patient cohorts and samples. So once you know what is the question that you're trying to address, it may be quite easy to figure out what are the right patients who should be enrolled in your discovery or replication studies. But it might not be quite so evident to know which are the right controls for the study. So that comes back to the fit for purpose. Once you know the fit for purpose, you have a better idea about what the right control patients are. So let's say we figured out the cohort. Now, what are the samples that we will be collecting? Because even within blood samples, you can collect them in so many different kinds of tubes, they may serve different purposes. So for example, plasma would be ideal for proteomics, serum for metabolomics. Buffy coat may be good for epigenetics, and blood stored in packaging tubes for transcriptomics. So you have to kind of figure out what's the best solution for you. Also, there is other bodily fluids or tissues that could be collected that needs to be figured out early on. And so one of the challenges could be around enrollment. It's quite it's time consuming, and it is quite expensive. So searching for researchers that have similar interests may lead you to finding out that they have a data or samples that they're willing to share with you. Or you could join forces to enrolling the patients together to share the cost. There is also other places that you could look for data or available samples, such as biobanks, that if you apply for, you may be able to obtain samples. Or there is a large data repositories, especially for omics data, such as Gene Expression Omnibus and others, where you would be able to access data, at least for a pilot study, so that you can do your initial analysis in it and know to have more confidence in developing your biomarker process. The other thing is, once you decide to enroll yourself, you may realize that the center you have access to doesn't have enough patients, so you have to enroll at multiple sites. Also, enrolling at multiple sites is quite important when it comes to replication, because you want to make sure that the biomarker you're developing applies to a wide range of patients. Now, another challenge could be around heterogeneous patients, especially with the complex disease that many of us are studying. The patients that we need in our cohorts are quite heterogeneous. If we are lucky and the signature of the question is quite strong, you may still be able to find good biomarkers. However, if that's not the case, you may have to focus on subgroups in which the biomarker has a strong signal and could be applied to. And the last challenge in this patient cohort and sample area is stakeholder participation. And this could come in more in the clinical utility phase, where patients or physicians may not be willing to participate. So involving them early to discuss the project, as well as to have input from them in designing the study, would ensure even higher patient safety and would get their buy-in. It would also help to work with Health Canada in a clinical utility study. Sometimes you even have to, but that also gives additional confidence to your stakeholders. But ultimately, especially in a pilot phase of the clinical utility study, you may want to allow the physician the option to use clinical acumen when they think that the biomarker is not giving the right response. Now, related to the patient cohorts is always clinical data that needs to be collected on them. And especially for a time course study, this could be sometimes the most expensive and most time-consuming part of enrollment, to collect clinical data. And especially now with the CST being at both PHC and VCHA, there is a lot more electronic patient data, even more than available before. And so the problem that Dr. Catherine Isaac came to us with, who is working in breast cancer research, was that they had a lot of PDF reports from both pathology reports and surgical notes that were in PDF format, and they usually had to do the extraction from these manually. And so she was looking for an automated way. And so together we had developed this natural language processing pipeline to assist in extraction of the data. So in a very simplistic way, how does this happen? The PDF is the input document. The text is extracted from there. And for this specific work, we were looking for a section of the report, and then extract the data using natural language processing, and then put it into a template, an Excel template that she had provided. So the reason for that was because she had also asked two medical students to do the extraction manually, independently, because we wanted to see how the manual and the automatic extraction compare. And we had found that the accuracy was high at 96.6%, pretty good for saving so much time in extracting the data. So now we're currently working with her to extract data from those more unstructured sections of the reports that require even more complicated natural language processing. Very exciting project. The second category of challenges is related to data. And even though I have here the omics data, it could be data coming off the clinical instrument and other data types. So it could be that you have a favorite platform, let's say it's epigenomics, but it may not be right for the clinical question. So it's important to check the literature ahead of time to see what others have found related to your question of interest and which sample and data types seem to be most suitable for the question. So even once the question or the platform is suitable for the clinical question, you may have a problem with not being right for the clinic. And so it's important to look for platforms that have the best way forward in clinic. Now, let's say you have done your discovery and replication successfully. Now it's time to move on to the clinical assay. And you may realize that the clinical assay is not able to provide the data back as fast as it's needed for clinical use. And so one would look for ways to improve the assay. And also another way would be to streamline the data delivery back to the physician. So around this ways of improving the assay, I have an example on the Genome Canada funded COPD project that was led by Dr. Sin and Eng. And once we had a diagnostic COPD exacerbation assay, we had gone to Dr. Maury DeMarco to to discuss ways to moving this into a mass spectrometer that is in the clinical laboratory. And we soon learned that this mass spectrometry would need an overnight digest, and that would not be usable in an emergency department setting. And so we needed a solution that had a rapid digest method. So Dr. DeMarco and her postdoc, Dr. Zhang, had set out to test different digestion methods. And they had found one that by 20 minutes was just as good as if you had an overnight digest. And so that meant that all of a sudden, from a 24-hour assay, we went down to a 2-hour return time to the emergency physicians. Pretty good. All right, the last category that I'm going to be covering are around intellectual property. And many of you may wonder, why is she talking about intellectual property? What does that have to do with anything? Well, when you get to the last phase of clinical implementation, you may need a commercial partner who will be definitely interested in having a patent on this biomarker process to make sure that they have freedom to operate in the space. Or you may decide to commercialize it yourself, but even then you will need funds from venture capitalists or others that would be interested in seeing that you have patent on the biomarkers. And so now that we established it's needed sometimes, you have to figure out when is the best time to file these patents. And so there is in academia this big push, to publish as early and as soon as possible. However, once you have a public disclosure of the biomarkers, either through a paper or a presentation such as this, those biomarkers become public knowledge and they can no longer be patented. So you have to balance this with the fact that you should file once you have more confidence in the biomarkers, such as after you replicated it or potentially even after the assay has been developed. So you have to balance this to figure out how and best to file in your specific case. Now, what are you filing really? First, the biomarker features, any unconventional methods such as technology platform or measurement methods should be included that make it unique. Also, the practical application of the assay or diagnostic method. So for example, how the clinical decision-making will be affected as a result of the biomarker. Will there be a drug therapy that's administered after? Would there be a procedure that is required if the biomarker is positive? And so all of those should be included. Where do you file? For this question to be answered, you have to think about what are the likely markets that you would want your biomarkers to be implemented in, and you would file the patents there. I have a link here to the UILO UBC office, and they have a lot of patent-related FAQs if you're interested to learn more about it. Now, as an example, I have listed here for the same COPD project for the diagnostic COPD biomarkers, we had filed the first provisional application 5 years ago. And within that, we had 1 year to file the PCT or Patent Cooperation Treaty application. Once that's filed, then you have 18 months to nationalize in the countries that you're interested. We had decided to nationalize it in Canada, the US, and in Europe. And so that happened 3 years ago, exactly. And so about a year or two after the nationalizations were submitted, we started to get office actions from the US and Europe. And after a lot of going back and forth with the patent officers, we finally had our first US patent approved just this month. So as you can see, this is a very long process, 5 years. And so take that into account as you're developing your biomarkers as well. So what other challenges could be that were not discussed in more detail? Some of them could be around the demand analysis. You have a specific question that you're just dying to study. And when you talk to the expert, they say, well, actually, there is not truly an unmet clinical need around it. Or after you talk to health economists, they may say, there is no socioeconomic to that. Or you think more about it, there is no path for clinical implementation. So that is so important to figure all of those out very early on so you can pivot to a question that would have a lot more impact. So I'm not covering challenges because the first two related to discovery and application have been discussed prior. And Jiyoung will talk about more around analytical development and clinical validation. But I want to mention things around clinical utility that you may see that there is a lack of funding because funding agencies think, oh, you're so close to having this commercialized. Well, you should foot the bill and try to fund this yourself. Or you may have issues in obtaining a Health Canada exemption if needed for your clinical utility trial. And as I mentioned before, in the clinical use, you may find that there is no commercial interest for any partners, or you have issues with reimbursement, or unless you involved your physicians early on, there would be slow or no uptake when you want to commercialize it. And so it's very crucial to start engaging your stakeholders, including the development and technology partners early on, so you don't run into these challenges. And in the last slide, I just wanted to drive it home that I'm hoping that I was able to illustrate it, but I just want to summarize it, that in order to develop fit-for-purpose biomarker tests, one needs expertise from many different areas, from clinical science to biomarker science to data science, as well as translation and implementation. And while within our center, we have expertise in many of these areas, we did not have a lot of expertise in the translation and implementation side. So we had to bring in experts. And so collaboration is the key for successful biomarker development. And with this, I'll hand it over to Jiyoung.
Okay, thank you, Zsuzsa. Thanks everyone for tuning in today. My name is Jiyoung Kim, and I'm a diagnostic test development scientist at Proof. As the previous talk illustrated, developing a biomarker is a long process with many challenges. So in this part of the talk, I would like to share with you on our experience of developing a diagnostic test called Heartbeat. As the name of our test, Heartbeat, suggests, the clinical problem we focused on was in areas of heart or heart disease, specifically in heart transplantation. Currently in Canada, heart transplants are monitored by regular biopsies, which means a procedure to take a small sample of the heart muscle is done between 12 to 15 times for a typical patient in the first year following heart transplantation. To show you what heart biopsies involve or what it looks like, I actually brought a short clip to share with you. This probe here is called bioptome, and that is what the surgeon will use to clip heart tissues. And the common access site for heart biopsy is through the jugular vein, which is right in the neck area. So after the patient is prepped, the jugular vein puncture is performed under local anesthesia. So the patients are awake, fully awake actually during this time, and all throughout the procedure actually. So when they're done with the procedure, they can get up and move around. So here, a sheath is being inserted, and this is needed to be able to for the bioptome to be inserted shortly. So what's being inserted right now is the bioptome. And at this point, the surgeon will use an echo to guide the bioptome to the right ventricle where the heart tissue is taken. So there is one piece of biopsy. And typically, they will harvest between 4 to 5 pieces of myocardial tissue. As you can probably imagine, patients do not enjoy getting biopsies. And I can personally attest to this when I go down to radiology here at St. Paul's to collect blood samples during scheduled biopsies. You can tell that this is clearly not an enjoyable thing to do for the patients. Also, while a biopsy procedure is fairly safe for the most part, it can still involve physical risks like infection. And biopsies are expensive. Each biopsy costs around $3,000, which adds up to about $40,000 per patient in the first year alone. And unfortunately, there is no alternative to these regular biopsies for now. So the question we asked at this point was, can we replace biopsy for monitoring acute rejection? Imagine transplant patients to go to their doctor's appointment for a checkup. Instead of making them go through biopsy procedures, we can take a little bit of their blood, take it to the lab, measure biomarkers that can tell the rejection risk for individual patients at that moment. The doctor is given the results, which is then used to counsel the patient for further clinical action. And this is what we exactly wanted to do. And to sort of bring this back to the unmet clinical need that we discussed in earlier slide, considering that there are over 200 cases of heart transplant per year in Canada and over 2,000 in North America, and each patient undergoing 12 or more biopsies in the first year alone, if we can find ways to replace biopsy, or at least some of the biopsies, with something less invasive and more cost-effective, it can alleviate both the emotional and cost burden on patients and the healthcare system. So we began this journey by trying to figure out what to look for. We considered many types of data, including clinical data, phenotypic data, various types of omics data, and using data analytics methods, some of which were introduced in our earlier seminars, part 1 and part 2. Initially, we were able to come up with a handful of gene sets or biomarker panels that could discriminate rejectors from non-rejectors based on their expressions in whole blood. Subsequently, we refined our panel to a 40 gene set, and again, evaluated this unified panel on patient samples to finally select best performing 9 genes, which are shown on the right in the black circle. This test is currently implemented on a nanostring platform that runs on this microfluidic device. I will talk more about the biomarker performance and assay characteristics in a few slides. And at this point, I thought it might be interesting to talk a little bit about the biology behind these 9 genes, because after all, these biomarkers probably came up in our work because in some way they are connected to the biology of immune rejection. So here, what we did was we took a list of genes that were interactors of the 9 heartbeat genes from various databases, including protein-protein interaction, from literature based on pathway information, and we found that the signals from the heartbeat genes and their interactors seem to largely come from CD4 positive lymphocytes and neutrophils, which is in line with what we know about the key roles that CD4 positive T lymphocytes play during acute rejection and the downstream events that end up causing organ damage by macrophages and other innate immune system components, including neutrophils. In our discovery work, which was largely done on microarrays, the heartbeat panel showed an AUC of 0.67 with 91% sensitivity and high negative predictive value of 99%. The negative predictive value describes the probability of the patient not rejecting given the test comes back negative. Recently, we were able to replicate the test performance in a new cohort of 160 subjects recruited from 8 sites across Canada. For this work, which was done on a nanostring platform, we saw an improvement of test characteristics of 0.73 AUC at 89% sensitivity, and again, high negative predictive value of 99%. So building on this strength of our test, heartbeat became a rule-out test for acute cellular rejection. After we had completed our replication work, we built this assay into a test that can be used in a clinical setting. And to do this, and this sort of goes back to some of the points that Zsuzsanna illustrated in her presentation, we evaluated a few technologies before finally choosing nanostring. And the factors we included things like clinically meaningful turnaround time, ease of uptake. So for example, the pathology reading of heart biopsies are usually completed within 24 hours after the biopsy is collected. So it was critical for us to choose a platform that will give us similar or better turnaround time. Also, we wanted to choose a platform that was already being used in clinical laboratories because that will reduce the test uptake cost for the clinical labs down the road. And also to make sure that the platform that we're choosing is appropriate for developing a clinical test. In the case of nanostring, they have a FDA-approved breast cancer panel called Prosigna that runs on the same technology. So after choosing the platform, we then developed the heartbeat clinical assay. And this process involves establishing various parameters about the assay that will help us understand how well and how consistently the assay performs. As an example, one parameter we needed to establish was how much sample to use. And this obviously depended on many factors, including how much RNA was available in the collected sample to begin with, or how sensitive our instrument was in detecting all 9 genes. So here, on the left is each gene's linear range, which is the range of concentration where the signals are directly proportional to the concentration of the biomarker in the sample. And you can see that it's not all the same for all 9 genes. So it would be important to pick a sample input where all 9 genes show this type of linear behavior. And here I'm showing 12 genes instead of 9, and that's because the heartbeat panel includes 3 housekeeping genes that we use to normalize other genes. On the right is the detection limit of our instrument, where you see the instrument losing signal or showing high level of noise at high or low extreme ends of input, respectively. So it would be best for us to use input ranges right in the middle. Based on this assay development and characterization work, we found that heartbeat assay had a very low CD, or coefficient of variation, of around 5%, and that is at individual gene level, with a low standard deviation of 0.02, and that refers to the heartbeat score, which is a single number that this assay outputs. And the score is on scale from 0 to 1. And I also want to mention that this assay has a short hands-on time of less than 30 minutes once RNA is extracted, and a fast turnaround time of 24 hours, which are fairly useful features for a test that is intended for use in clinical settings. Importantly, we also conducted a pilot clinical validation on 32 new patient samples and found that the panel performance to be highly similar to what we've seen in our replication work, which was very reassuring. So summing up everything I presented here to you today, we believe this is what heartbeat can offer. Instead of everyone getting biopsied regularly, heartbeat can be used to select patients who will not benefit from a biopsy because they are stable and their graft is doing fine. We estimate that we can eliminate around 40% of unnecessary biopsies in the first 12 months post heart transplant by using heartbeat assay. And now we're conducting a multi-center observational clinical trial for evaluation of this test. We will be collecting rejection and non-rejection samples from patients enrolled from these 4 participating hospitals. We will also be following patients in the first year post-transplant and collect samples during that time. So we'll be able to generate time course data, which can help us understand what happens before, during, and after rejection. And also, we had previously identified actually some protein and miRNA candidates as part of our discovery work, which I was not able to go into detail for today's talk, but we plan to evaluate if the current heartbeat test can be further improved if we can combine the current mRNA targets with either the protein or the mRNA candidates. So this concludes Proof's 3-part webinar series. Thanks for being here today. For those of you who missed our previous talks, as Karen spoke about earlier at the beginning of the session today, you can find the recorded seminar on YouTube on Proof Center's channel. You can type in Proof Center with a TRE in the search bar on YouTube and it'll pull up our channel.